The overall goal of this procedure is to track protein movement by expressing a single protein bearing two different fluorophores in two separate cell lines and fusing these cell lines together. This is accomplished by first transfecting plasmids bearing the gene of interest fused to either of two different fluoroprotein genes into two separate cell types. The second step of the procedure is to mix the cells together and allow each cell type to accumulate protein. The third step of the procedure is to inhibit protein synthesis, then fuse the cells using polyethylene glycol. The final step of the procedure is to visualize protein localization via fluorescence microscopy. Ultimately, results can be obtained that show alteration of protein localization or trafficking behavior through fluorescence microscopy.
This method can help to answer key questions in the cancer biology field, such as how do discrete protein functions and behaviors change as cells become transformed?
Though this method can provide insight into transformed cell-specific changes, it can also be applied to other studies, such as cell differentiation, developmental biology, and cell signaling.
Generally, individuals new to this method will struggle because optimization of transfection conditions as well as fusion conditions will be required for each different cell type.
The first method demonstrated in this protocol for the introduction of plasmid DNA into cells is the nucleofection method, which uses Lonza nucleofector solution. To begin, wash cells that have been recently passaged and are in logarithmic growth with phosphate-buffered saline, PBS, and harvest them by trypsinization. Once the cells have lost adherence, neutralize the trypsin with the addition of serum-containing media and collect cells by centrifugation at 1500 G for 5 minutes. While the cells are being centrifuged, add sterilized cover slips to a 6-well plate in the tissue culture hood. Then, pipette 1.5 milliliters of complete culture media into each well. Cover and move the plate to the cell culture incubator to equilibrate. After centrifugation, move the tubes to the hood, aspirate the supernatant, and resuspend the cell pellets in a minimal volume of PBS. Next, count the cells and aliquot the volume of cell suspension to sterile microcentrifuge tubes that would provide approximately 5 × 10⁵ cells per sample. Centrifuge again at 1500 G for 5 minutes. Once the centrifugation is complete, place the tubes in the cell culture hood. Aspirate the supernatant and carefully resuspend the cells in each tube with 100 microliters of room temperature nucleofector solution. Then, add 5 micrograms of plasmid DNA to each sample and transfer the sample to a nucleofector cuvette and cover. Take care to avoid air bubbles. Insert the sample cuvette into the cuvette holder, select the appropriate nucleofector program, and initiate it. Selecting the most appropriate program may require prior testing to establish optimal transfection efficiency with minimal mortality. Then, plate the cells into the 6-well plate in either mixed populations or separately as controls. Return the plate to the incubator and allow the cells to recover for 12 hours. The second method demonstrated for transient expression of plasmids is transfection with QIAGEN affecting reagent. To begin, add 0.8 micrograms of each plasmid DNA sample to 200 microliters of EC buffer in a sterile microfuge tube. Then, add 6.4 microliters of enhancer reagent to each sample, mix briefly by flicking the tube, and incubate for 2 minutes at room temperature. Following this brief incubation, add 20 microliters of affecting reagent to each sample. Mix each tube by flicking and incubate for 15 minutes at room temperature. During the incubation, prepare the 2 cell lines to be used in the experiment for transfection. The cells should be recently passaged and in logarithmic growth at approximately 80% confluence. Wash the cells once with PBS and add back 1.5 milliliters of fresh DMEM with 10% FBS. Now that the DNA transfection reagents have incubated for 15 minutes, add 1.2 milliliters of DMEM plus 10% FBS media to each sample. Mix by pipetting and immediately transfer dropwise approximately 700 microliters of the complexes to each of 2 wells in the 6-well plate. Mix by swirling the plate. Allow the cells to transfect for at least 3 hours in the incubator. In the cell culture hood, place 1 sterilized cover slip per well in a 6-well plate. If desired, coated cover slips may be used to enhance cell adhesion. Next, Wash the transfected cells twice in PBS and harvest the cells by minimal trypsinization. This is accomplished by adding 100 microliters of trypsin solution to each well, agitating the plate to ensure complete coverage, then immediately aspirating the residual trypsin. Neutralize the remaining trypsin by resuspending the cells in 1.5 milliliters of fresh DMEM with 10% FBS. Plate the cells in either mixed populations or separately as controls in the 6-well plate containing cover slips. Allow the cells to recover for 12 hours in the cell culture incubator. Regardless of the transfection method, the procedure for generation of heterokaryon fusions is the same. Following transfection, remove the culture media from the wells and wash the cells twice with PBS. Next, it is recommended to treat the cells with cyclohexamide to inhibit further protein synthesis. Add cyclohexamide at a concentration of 100 micrograms per milliliter in culture media to cover the cells and incubate for 2 hours in the cell culture incubator. After 2 hours, remove the cells from the incubator and wash the cells twice with PBS. To initiate fusion, cover the cells with a solution of 50% polyethylene glycol 1000 in serum-free DMEM and incubate for 125 seconds at room temperature. Remove the PEG solution and wash the cells with PBS to remove residual PEG. Then, add 2 milliliters of DMEM plus 10% FBS warmed to 37 degrees Celsius. Incubate the cells for 18 to 24 hours in the cell culture incubator. Following the 18 to 24 hour incubation, retrieve the plate from the incubator and wash the cells twice in PBS. Next, pipette 1 milliliter of 4% paraformaldehyde in PBS to each well and gently agitate for 15 minutes. During the incubation, prepare the microscope slides to be used by adding a single drop of mounting medium containing DAPI. Then, wash the fixed cells twice with PBS and carefully remove each cover slip with small forceps, wicking away residual PBS on a piece of tissue before placing it cell side down on a microscope slide with mounting medium plus DAPI. Remove the excess mounting medium from the slide with tissue paper and seal the edges of the cover slips with nail polish. The cells can now be visualized by confocal or epifluorescence microscopy. In this flowchart, the heterokaryon method for analysis of cell type-specific localization of proteins is shown in 3 basic steps: transfection of the plasmid expressing a fluorescently tagged protein, cell fusion with polyethylene glycol 1000, and fluorescent microscopy imaging. As can be seen in these epifluorescence images, primary foreskin fibroblast self-fused controls expressing GFP-tagged chicken anemia virus VP3 protein show primarily cytoplasmic localization of the protein. In the non-small cell lung carcinoma line H1299, self-fused controls with the same VP3 protein tagged with DS-red show primarily nuclear localization. Heterokaryon fusion of the primary foreskin fibroblast and H1299 cells results in the introduction of transformed cell material to the primary foreskin fibroblast and causes a change in localization of GFP-tagged primary cell-derived VP3 to the nucleus.
While attempting this procedure, it's important to remember that success will be highly dependent on optimizing transfection efficiencies in the particular cell types being used, as well as optimizing fusion conditions for these particular cells.
Following this procedure, other methods like FLIP and FRAP can be performed in order to answer additional questions involving real-time analysis of localization dynamics.
After watching this video, you should have a good understanding of how to assess cell type-specific protein localizations by introducing fluorescently tagged proteins and generating cell fusions.